Hi everyone, this is Mrs. Ortega here. I am excited to get going on some new information with you as we have finally tackled in previous exams and chapters evolution and plants, and now we get to move on to animals, which is really fun and really exciting. And so I hope you will enjoy the next set of chapters, of chapters 6, 7, and 8 as we look at animals. Invertebrates, those without a backbone, and vertebrates, those with a backbone. And so as we go through these chapters, I will try to be diligent on making sure we cover everything you need to know for the exams, as well as notifying you of any changes and anything you don't need to know for the exam. So this chapter here we'll get started on today is chapter 6, and we'll cover invertebrate diversity. So in this image here you can see a bunch of different insects, and so that's what our book starts out with, that there is a vast diversity of insects, and they have a wide variety, as you can tell, of shapes and sizes, habitats they live in, diets of food they eat, mating habits, and just many other characteristics that they vary in. And so all of these animals, even though they have a wide variety of different aspects, they are tied together by some common animal traits. And so those are things that we will look at today. So looking at chapter 6, there are kind of two big ideas here. First, animal evolution and diversity. So how do all of the animals connect? How do they, what are properties that they all share, as well as how are they diverse? How are they different from one another? How do we group these as well? And then we'll go on to invertebrate diversity in particular, looking at those animals without a backbone. So here we'll start with animal evolution and diversity. Here asks us, what is an animal? Now that sounds pretty easy to define, but if you think about it, we have animals that have fur, we have animals that have feathers, that have 4 legs, that have 2 legs, that have beaks, that have mouths, that have teeth, all those different things. It does make it hard to classify what is an animal. So animals are eukaryotes. So they have eukaryotic cells. These cells have membrane-bound organelles. All right, so no bacteria. No more bacteria for us in this section. So animals are also multicellular heterotrophs. So we are made up of many cells, and we will eat by ingesting things from the environment. So heterotrophs are taking in nutrition from the outside environment. And then we also lack cell walls. So that distinguishes us from the plants, and that plants have plant cell walls where we do not. So you'll want to remember those unifying traits of animals here. And so looking at other things as well, animals will eat food, and that process is called ingestion. We'll take a look at that process of ingestion and digestion in the nutrition chapter. But here in this chapter, we'll see some embryonic development connections of all animals, as well as some general life cycles. And so these do have unifying features, but can also help distinguish them from other groups as they have some subdivisions. And so here, looking at our example of this rock python digesting a gazelle. This is a form of ingestion, the eating of food. In particular, this is bulk feeding that we'll learn in the nutrition chapter. This is a large bulk feeding that will probably take this python a couple months to digest, if I'm remembering correctly. And so looking at embryonic development, so the development of the embryo to the adult also shows us some unifying features of animals. Sorry for the slight interruption there. Most of you hopefully did not hear that background noise, but if you did, I apologize. I do have a very loud, obnoxious pet bird, which is one of the reasons why I've been filming these, pre-recording these, so I make sure she is quiet or outside during this process. Okay, where was I? We were here with animal embryonic development. Okay, so starting with the embryo, you want to remember what the embryo is, which is the zygote as well. Common name for the whole process will be embryo, but the zygote is going to be the fertilized egg, which is here. So the fertilized egg is now a zygote, and that develops into the early form of the adult, which is the embryo. But this zygote has to have come from the sperm fertilizing the egg. And so once that happens, then development happens, where the cell will divide and reproduce more cells, and those cells will divide. And so here's an 8-cell stage, very early stage. Then the next stage is called the blastula. This is a hollow ball of cells. And so you can think for this section, blastula, let's go with this one here, starts with the letter B. And B is also the first letter in ball. So a hollow ball of cells. And then moving along to the gastrula, that will be the next stage of embryonic development. And so the gastrula is going to be very similar to the blastula, except that it is going to start forming a new layer. And so that new layer is done by pinching inward here. So that little portion that's pinching inward is going to form a second layer. That process is called gastrulation. And so gastrulation is coming from the gastrula, which is a ball of cells that is going to have more than one layer. And you can see that in the later gastrula cross-section here. There's development of different dermal tissue that we'll see, and then some internal sac there. And then as that develops, it will then go into what we'll call larva. And so for this example, it's a sea star. So the sea star or starfish larva is going to have a different body plan than the adult. And so that's what a larva, it's going to be a juvenile with a different body plan. And then the juvenile is going to have the same body plan as the adult, but it's not going to be capable of sexual reproduction yet until it undergoes completion of development into an adult. And so for us, we humans have obviously the same body plan. Once they are born to an adult, it just develops over time. And so we would be, instead of larva here, it would just be a juvenile in biology terms. And so on the next slide here, we have those terms defined, that blastula and gastrula, and then the dermal layers that we'll learn in the development and later slides are also defined here. So in terms of studying, I'll try to go through these as we star them as we go along. So this, along with this slide here, and this slide here. All right, so looking at animals in terms of fossils and evolutionary history, you would go back about 560 million years ago to the Ediacaran period. This period had animals that were soft-bodied, that had varied in shape and length. And so as time moved on to the next period in time, the Cambrian period with the Cambrian explosion, that had a rapid diversification of animals about 535 to 525 million years ago. In this period, the Cambrian animals had harder body parts like shells and spikes. And so you can see these in these examples. This one here, the Ediacaran fossils, that they have soft bodied, and so their imprints here, as you can tell, do have some vertebrae, possibly, or some vertebrae-like structures, but overall, you can tell they're mostly soft tissue. Whereas here, they have hard spikes in this drawing rendition of the Cambrian period, and the hard shelled parts. And so looking at animal diversification, there's about 35 or phyla. Some systematics disagree on the precise number, and only one of these phylum includes vertebrates that have a backbone. And so roughly 96% of the animals are invertebrates, animals that lack a backbone. As we'll see here, most of the classification of the invertebrates you'll be familiar with, and some of them are probably entirely new to you, but overall most of the classification of animals are actually invertebrates, not vertebrates. Our checkpoint question here says, what are two major differences between the fossil animals from the Ediacaran and the Cambrian periods? They had soft bodies here. And then the Cambrian did not. So they had hard body parts. Okay. So looking at animals in terms of body plans, it does vary greatly, but we'll see some unifying features like symmetry, tissues, embryonic layers, something called a body cavity, and then embryonic development details. So all these help unify animals, and so that is, I believe, something you'll want to understand. Let me check here. Yeah, this is something you do want to know. Alright, so first, type of symmetry. Symmetry is usually one of the easier ones to understand, and that radial symmetry, no matter which way you cut the organism in half, it's going to look the same on both sides. Here we have a sea anemone as an example. Another example would be a starfish or a sea urchin. In terms of maybe more relative terms, like a pizza would be something you can cut in half no matter what, and it'll look the same on both sides generally. So that would be a good real life example. Whereas the bilateral symmetry, you can only cut in half in one way for it to be equal on each side. And so here we have the lobster, and you're cutting it in half from like left to right side, and so that if you were to cut it in half along the torso, that wouldn't work, and so you wouldn't have symmetry on both sides. And so we have bilateral symmetry. If you have an imaginary line down the middle of our bodies from head to toe, you'll have equal symmetry on each side. So left and right hand, left and right foot, and so forth. All right. Looking at embryonic development, as we continue that topic here, the blastula forms the gastrula, and then from the gastrula it pinches inward, forming different tissue layers, right? And so here in this example, we have the first opening in the embryo and gastrulation forming a second tissue layer, which is shown here and color-coded for you. The ectoderm is the outermost layer in blue. This here is the outermost layer. Then the endoderm is the innermost layer, which is in yellow. And then the mesoderm, which is up here, will be in red there. And so those are the three embryonic layers. And so if we go back up to that same slide here, you'll see what they give rise to. So the endoderm gives rise to the digestive tract and other hollow organs. So you think any hollow organs in your body, like the stomach, mouth, esophagus, small and large intestine, colon, all of those are hollow in the center. And so those are from the endoderm of the embryo. Then the ectoderm is the outer layer. It gives rise to the epidermis, which is our skin, and then the nervous system. So brain, spinal cord, etc. So mesoderm is the middle layer, and that gives rise to everything else. So muscles, bones, the lower layer of the skin, the dermis. Most organs that aren't hollow are from the mesoderm. And so going back, let's look at the tissue layers. So in the three tissue layers here, there's a further division for development for animals, and that they are called the protostome and the deuterostome. And that the protostome, the first opening, becomes the future mouth. So the first opening of that gastrulation process becomes a future mouth, whereas the deuterostomes, that first opening becomes the future anus. And so if you were to try to guess which one we would be a part of, we are part of the deuterostomes. And so while these layers in particular are not something like you don't need to memorize this page or anything, but you will see these terms later on throughout the discussion of animals. And so that's why I like for you to understand it now as we go through. And so looking at this next one here, the next feature of animals that's studied is the body cavity, the presence of one, the absence of one, the formation of it. And so body cavity is also known as the coelom. It's spelled weird, but it's pronounced coelom. And so this is a body cavity. A body cavity is simply an opening or empty region within the tissue of the animal. And so looking at a true body cavity are examples of earthworm. This earthworm here, if you were to cut it across stem-wise, you would see all the different layers of cells. First, you see the ectodermal layer in blue, and then the mesoderm layer in red, and then lastly the endoderm layer in yellow. And so in that mesoderm area, you can see two body cavities that are lined on either side with mesoderm tissue. And so that's a true body cavity. So that's something you'll want to understand, that a body cavity is an opening there of basically empty space within the internal tissue. All right, so biologists will use all the information about body plans, about the fossil record, along with molecular data like DNA sequences, to group animals into different phylum and different groupings for phylogeny as well on the evolutionary tree. So this is the general accepted phylogenetic tree for the animals that we're looking at here. And so if you start Beginning, there's ancestral colonial protists. So this is, you remember the tiny eukaryotes that we saw in chapter 4. So that was the ancestor that then eventually adapted no tissues to become what we know as the sponges here. So sponges were the first to evolve and were diverged. And then going down the other way here, you see they do develop tissues, and then the group called eumetozoans include all the ones you see here following just everything but sponges. And so the first ones to diverge with radial symmetry are the Cnidarians. So as you go down like that, you can see the groupings along with the names of those groups. And so a question asks us, what shared derived character separates the Bilaterians from the Cnidarians? So Cnidarians are here, and Bilaterians are right here. And so what character is bilateral symmetry? All right, so we are now on the invertebrate diversity portion. This portion, we go through examples of invertebrates in different phylum. And so as we look through these, I will try my best to make sure I point out which structures or features to understand and know for the exam. And so keep that in mind as we go through. First one, sponges. Sponges are very simple, porous body that they will filter feed through their environment. They do not have true body tissues, which is one of their unique features. They usually don't have any body symmetry, and most of them are marine, but some of them can be found in fresh water, but all of them will be found in water. And so they have these unique cells called choanocytes. They are, it's spelled here for you, and these cells will filter the water for food. And so we'll look at the structures of choanocytes and how they actually do that process for sponges. Another cell that sponges have are called amoebocytes, and they will kind of hold everything together through making skeletal fibers called spongin and spicules. And then they also digest food that's obtained by the choanocytes. So here we can see some examples of sponges. So as your face sponge, the Sypha sponge is one we'll look at in lab. In lab next week, we will see a lot of sponges and Cnidarians. And so also here, purple tube sponge. So there are different classifications of sponges that we'll see in lab. But the general process is shown here in terms of filter feeding. The water is going to flow in through the sides of the sponge, and the openings are called ostia. We'll learn in lab. These are called ostia. They're openings that allow for the water to come in. Here they just note that it's a pore. So really it's an ostia. And so as the water moves in, then you see the nice little choanocyte cells that look kind of like a round end with then like a weird collar on the other end. That is a choanocyte that's going to have a flagella in the center that's going to propel and go rotate in circle form. So in circle form, it's going to spin, 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 and that's going to propel water in, and then the little collar part, the little appendages on the sides here, those will collect the food. And so as that happens, then that water gets propelled to the center of the sponge. The center is called the spongocoel. Just sponge. Seal. It has a unique spelling as well. And so that's the sponge cavity, right? Seal coming from cavity. And that's supposed to be an E, I'm sorry. There. And then the water goes to the center of the spongocoel, and then it will flow outwards. The opening that, or the pore that allows it to leave is called the osculum. All these terms we'll see in lab. That's why I point them out now. All right. All right, so why is it thought that sponges represent the earliest branch of the animal kingdom? Well, it's due to their factors that they lack true tissues, right? All the animals, other animals we'll see have true tissues. Their choanocytes resemble something we've seen before in the Protus group. The Protus group had a unique structure in one of the ammo-like Protus groups that was closely resembled that choanocyte that you see here. Alrighty, so looking at the next group, the Cnidarians, these are radial animals with tentacles and stinging cells. So Cnidarians are phylum Cnidaria. They have radial symmetry. They also have two true tissue layers. They have a gastrovascular cavity, which is a structure we will look at. And then they have two body forms that they're most famous for. The polyp form is a stationary form, and the medusa is a mobile form, and we'll look at those. And so here are some examples of common cnidarians. The marine jelly is a medusa form of a Cnidaria group. Sea anemone is a polyp form, as it's stationary. It has is not going to be mobile. And then the hydra alternates between being stationary and being mobile. And so that is another example. And so all these have tentacles with stinging cells on the ends of them in different regions. And so looking at those tentacles and stinging cells, on the tentacle of the hydra zoomed up here, that tentacle has regions of normal cell, and it has regions of the stinging cell, which is called the synocyte. Right here, synocyte. And so on the synocyte itself, they have a region called the capsule that has a coiled thread, or what we call a trigger, on the outer portion sticking out. And so when that trigger touches prey tissue, it's going to discharge that thread that will then be inserted into the prey tissue and cause that stinging sensation. In terms of a video for the mobile Cnidarian, so we have this jellyfish video here. Okay. And then for hydras, hydras are an organism we'll see in lab, so I like to kind of preface it now, that it has a unique body of a region called the basal disc that will attach to a substrate when needed. And then they have this kind of stalk body, and so at the end of that stalk body, in the center, there's a mouth. So they'll swallow food through the mouth, and then that will enter what we call the gastrovascular cavity, something all Cnidarians have. And that gastrovascular cavity will break apart the food for not only nutrients, but for vascular needs like oxygen and carbon dioxide gets released. And so as that creates waste, that will then be expelled out of the mouth, and then it'll be ready for some more food. And so to capture food, it will use its tentacles that will sting different small organisms. For the hydra, they are very, very small. You can see them just barely with your eyes, but most of the time in lab we would have some live ones that you can see under one of those large stereoscopes. And so here is a video of a hydra eating a small organism called a Daphnia. So let's take a look at that. So there he's grabbing it, and now he's trying to swallow it. So it's about mid-swallow there. He's clutching on to that Daphnia pretty well and trying to take it into its body. And looks like it's completely swallowed now, and now it will break apart. So another unique feature for the hydra is that they have both means of reproduction. So they're capable of sexual reproduction as well as asexual reproduction. That's shown on here on the picture as well. They have a fully grown bud asexually growing on the side. And then they have the gametes needed for sexual reproduction as well. And then another example of the Cnidarians are the LCME, which is a polyp form. This is a stationary form. And so looking at the stationary form here, I believe this is a video. You can see clownfish like to live in sea anemones for protection from other larger predators. So what are the three functions of a Cnidarian's gastrovascular cavity? So they will be responsible for digestion, so the breaking apart of nutrients and spreading those nutrients to the other body tissue. Circulation, so transport of everything, so the nutrients we just discussed, as well as oxygen and waste like carbon dioxide. It also is responsible for physical support and movement. So the gastrovascular cavity will be an internal tissue, but still helping the animal in terms of support and movement. All right, on to the next group. And this group includes flatworms. So flatworms are the simplest of bilateral animals. The flatworm group for the phylum name is Platyhelminthes. This is a bilateral animal with no body cavity. They also have a gastrovascular cavity, and for one type of flatworm, it has a simple nervous system. And so this flatworm group consists of many different types of worms. One of them is non-parasitic, and that's the Planarian. Here, Planarians are the only group that's non-parasitic or free-living. So they, as opposed to the other group here, flukes and tapeworms are parasitic flatworms with different and complex life cycles. So in terms of flatworms, you can think two kind of subdivisions. One's a Planarian, that's non-parasitic, free-living, and the other group is a parasitic group that includes flukes and tapeworms. And so all flatworms live in either marine, freshwater, or damp terrestrial habitats. They do need that water in some form or another. Looking at our example here, this is a body plan of the Planarian. And so in the Planarian, this is non-parasitic. It's going to be the free-living species. It has one opening, and it's named the mouth. From here, food will be taken in, and then the food will go through the whole gastrovascular cavity, which is here in brown. As you can see, it spans pretty much the whole body of the Planarian. And so that circulates all the nutrients, the oxygen, the waste, and then expels it out back through the mouth. At the anterior end of the Planarian, you have eye cups or photosynthetic, or not light detecting eye cell region. And you also have nervous tissue clusters around there, like a very simple brain that has nerve cords running on each side, the right and left sides, all the way through the whole body. And so this Planarian is unique in that it's a very, very, very flat. And when you see these under the microscope in lab, which we'll get to that eventually, they will be very easy to see through the whole body because it's so thin. And the reason why it's so thin is that its skin surface does gas exchange. And so this gas exchange is taking out the middleman of the gastrovascular cavity for the primary responsible role for getting oxygen in and carbon dioxide out. And so it does it through its skin, and having a very flat surface allows it to do that efficiently. All right, compare that to your tapeworm, which is shown here. A tapeworm is going to be a very long worm, and the anterior end is what we call the scolex. This is the region where it's going to attach to its host. So the attachment point will be at the hook region, and then the suckers here will be responsible for attachment as well. And so as we look at the tapeworm, we'll see it has different kind of small segments that repeat over and over and over again. And so as the tapeworm gets older, it gets longer. And as the tail region gets longer, some of it's going to start to break off, leave the digestive system through the feces, and then that feces is now contaminated and it could infect other organisms and give them the tapeworm. And so the little units that break off are ripened eggs ready for ready to grow a new tapeworm essentially. And so the longer it is, the more offspring is going to eventually create. All right, so a checkpoint question here. Flatworms and Cnidarians differ in symmetry, with flatworms being bilateral will be the answer, and Cnidarians being radial. But the animals of both phyla have a gastrovascular cavity. So looking at that, flatworms, you can cut them in half from the head to tail region, and it will be symmetrical, and that's the only way you can cut it. Whereas Cnidarians here, a nice example of the sea anemone as well as the jellyfish and the hydra as well. If you cut them in half anyway, it's going to be equal on both sides, or for the most part equal. So that will be radial. And they both have gastrovascular cavities. So remembering gastrovascular cavity will break down the nutrients as well as the gas molecules, if you will, like carbon dioxide and oxygen. All right, on to nematodes. Nematodes are one of those we normally dissect in lab. I am still working on figuring out what we're going to do in place of dissections. Most likely watching videos of other people dissecting them is what we'll probably do. And usually nematodes is one of them, so I like to kind of go over that pretty well now. So nematodes are also known as roundworms, is a common name. They will have bilateral symmetry, 3 tissue layers, a non-living protective coat called the cuticle, just like in plants, that cuticle waxy covering layer. But for nematodes, this will be shed periodically. And then they also have a complete digestive tract with a mouth and anus. Some nematodes are free-living, and some are parasites. And so here we'll look at, in terms of characteristics, the cuticle, and that's pretty much it for the defining unique characteristics, that cuticle. And so you can see here in this left image that it is like a corduroy type texture. That cuticle, the outermost covering, looks like soft and velvety almost. That's going to be shed periodically as the nematode needs. The nematode is bilateral symmetry, or has bilateral symmetry. So if you cut it in half long ways, that's going to make it symmetrical. The blunt end, the rounded end here is the mouth, and then the tapered end here is the tail. So that's something normally we look at in lab and try to distinguish what side's the mouth and what side's the tail. And you look for the tapered end is the tail, the blunt end is the mouth. And then for nematodes, some of them are free-living and some of them are parasitic. And so the one example of parasitic here is infesting a large artery of the porpoise whale, but there are other nematodes that you might be familiar with. The first one that's parasitic will be heartworms. So heartworms, like for dogs, they will invade the heart. And so giving dogs a monthly treatment, heart guard prevention, will prevent these from being able to survive if ingested. And this is a type of nematode here. Another type of nematode is something called C. elegans. Oh, no. Go back. C. elegans. This is a nematode that's free-living, so it's not parasitic. And sadly, it is one of the most well-studied nematodes out there. So of all the nematodes, scientists picked the one that's free-living to dissect and to investigate. And so C. elegans is one of the few that's free-living, that's not going to be a parasite, and it's also a model for research in the scientific community. And so as they research this, they will see that they have a complete digestive tract from mouth to anus at the end there. They also have a liquid body filling that will be called a pseudocoel. This is something we'll see in lab. Pseudocoel that will bathe the internal organs and spread the nutrients that way. And then it has only longitudinal muscle going from the mouth to the tail here. And so that allows it to move from side to side. And so that's what you can see in this video here. Is the movement of the, I believe it's the C. elegans, yes, our poor free-living nematode that gets scientifically researched all too often. So you can see how it squiggles from side to side. That's from that longitudinal muscle going from head to tail. All right. And now we are on to the mollusk group. I'm going to just check in our time. Unfortunately, I have to pause for now, but I will make a second recording with the second half of this information for you. So hang in there, and I will get this going as soon as I can. But for now, we'll sign off. Bye-bye.